Early sponge fossils are certainly very strange when it comes to comparisons with modern groups. Some, however, are certainly stranger than others, and it has to be said that one in particular really stands out. Takakkawia is so bizarre in so many ways that you have to start wondering whether it's even a sponge at all, especially when you start noticing the connections, potential connections, to fossils that appear to be early ctenophores. Hello everyone, and thanks for tuning in for another instalment of Sponginess. I also just want to thank you for your patience in me responding to comments and queries quite slowly over the last week or so, because I've been away for another couple of days playing with the big microscopes. But also there's just so many people commenting now that it's actually getting quite difficult to keep up. So I am reading everything, and please do understand if I'm a bit slow to respond to a particular query or question that you have. I try to get round to any of those eventually, but it's getting a little bit more difficult now. There's so many more of you, which I'm not complaining about at all. So do stay. Right, back to sponges. As you know, unless you're completely new here, I'm doing a series on early sponge evolution, which is diving into the details. It's actually well beyond what you'll get in university textbooks, for example, or even a degree course, and a lot of it draws on our own papers over the last 20 odd years, where we've been challenging the traditional views, I think it's fair to say. I'm not trying to present our ideas as facts, because that's not how science works. But I can present our arguments and the observations that we've been making as openly as possible, and where a traditional interpretation is frankly ridiculous, can explain why and express that opinion along those lines. And that is exactly what we have here. So in the previous video in this series on the ascosponges, I explained, hopefully convincingly, why these Cambrian things just do not fit into demosponges where they've traditionally been put, meaning that the old assignment of the protomonaxenids had to be discarded. Even before I formalised that, though, there was one genus that I'd kicked out of the group entirely on the basis that it basically shared nothing with the rest of them, or indeed with any other fossil sponges at the time. That genus is Takakkawia. Even from the first descriptions by Walcott back in 1920, it was clearly very odd. Unlike any other sponge in existence, it had a series of fins sticking out round the side of it, 8 of them. So let me just read out the diagnosis. This is from 1920, when Walcott was trying to squeeze everything he could into existing groups, and in this case it was a demosponge that he was putting it into. So, slender, cylindrical, thin-walled sponge with its skeleton formed of vertical strands of long spicules, with vertical bands of delicate simple spicules embedded in spongin. Fine transverse spicules occur singly and in fine strands. So you read that, it actually sounds rather like a leptomitid, if you remember those. So you've got longitudinal spicules running in bands up the sides, and you've got transverse spicules, sometimes in bundles, running around it as well. That doesn't really do it justice at all. So even in the photographs, you can see that there is something very strange about this species, and that is these weird fins sticking out from the side. There is no other sponge that does something like this. But if you ignore those features and just combine the small monaxons with spongin, then you end up with demosponges. The evidence for spongin is that these columns of spicules sort of hold together really well, even when the sponge is decaying, and therefore it must have been bound by spongin, was the argument. Again, it's a question of looking at modern sponges, seeing which little bits you could pinch from different groups, sticking them together and say, well, spongin, demosponges. And that was the approach that was being used at that time. And we now know that most of the fossils that were sort of shoved into modern groups didn't really belong there. Okay, So Rigby redescribed it in 1986 in much more detail and gave actually detailed drawings of the structure. So this has expanded the diagnosis quite significantly, but hasn't changed a huge amount. So we have a reconstruction which at least truly acknowledges these strange fins that are sticking out from the side. And there are 8 of them. He noticed that. He said there's quite often 4, but that's only when you can only see half of it. So there are 8 of these fins. And then he gave a detailed description of and even a diagram of the internal structure, the skeleton of it, where you have these twisted rods of little monaxon spicules that flare up at the end into these little sort of rosettes, almost little sort of flower bud-like structures. And that was the next instalment. He still included them in demosponges, still considered these rods to be these bundles of spicules going vertically up to be composed of lots and lots of little monaxons, but twisted round in a sort of helix array. And then he mentions these blades sticking out, goes into some detail about how they're clearly very thin structures, sort of sheet-like things, and doesn't really get into interpretation or biological significance of those features. Just does at least acknowledge that they're there. So in 2004, there was the update of the Burgess shale sponges by Rigby and Collins, where they described new material had been found since then and added a little bit more detail. Not a huge amount. In fact, the only real addition to that is that, to the previous description, is that some of the transverse spicules he thought may be the rays of hexactins that were actually part of the vertical rods of spicules, and then there's a ray sticking out sideways. So suggesting there might be hexactin derivatives there as well as monaxons. But it didn't stop him putting them in the demosponges, just said need better material to make sure. But at least that is now making it fairly clear that it's very difficult to interpret this as a demosponge in all sorts of ways, because there is nothing like these fins in any modern sponge anywhere. Okay, so in 2011 I had the chance to work at the Royal Ontario Museum for a few weeks on the Burgess shale sponges, and one of the things that turned up from that trip was that I had access to all the Takakkawia specimens, and I couldn't resist spending a good long while on them because it's obviously such a crazy little creature. So I did, and it may well be that when Rigby was looking, he really was given the best, the most complete specimens, and I got lucky. I got the grotty ones as well, the ones that were falling apart, the ones that are disarticulating, and that was a revelation, because when you can see the individual components of them, you realise that these previous reconstructions were nothing like correct. For example, those twisted bundles of monaxons in the sort of helical arrangement going up the sides, nothing of the sort. There are no bundles of monaxons there at all. What you have instead is single, slightly curved monaxons, a single line of them which are overlapping each other by about half the length. And of course, if you have 2 curved monaxons overlying each other like that, you end up with something that superficially resembles a twisted helix. And that was what he was seeing, I think. But it gets worse than that, because rather than being a separate fin sticking out, those fins are actually also overlapping parts of these spicules. So the single rods actually support a sail-like structure sticking outwards from it, and those also are overlapping. So you end up with a fin as the overlapping structures of these sort of isolated spicules with a big organic sail sticking out from the side of it. And this is extraordinary. It's a really weird structure. But those sails, those sort of flanges sticking out, also have concentric ridges going out across them, which makes it look like there's many more spicules there than there really are. So when you start to see those in isolation, it's pretty clear what you're looking at, and you can start to reconstruct it very, very differently. those structures around the osculum that you described as sort of like the untwisting of these bundles into a sort of flower bud-like thing were also revealed, and they are in fact very similar to the things making the phalanges, only different. So you have, again, very simple spicules. At the top of these fins there is a single sort of monaxon spicule with, again, a sort of sail coming off it, but of a different shape, so it's clearly the terminal one of that sequence, and it forms a sort of strut surrounding and radiating out from the margin of the osculum, the hole at the top. But between those 8 there are also smaller spicules along the edge, which are not monaxons. They actually are triaxons, as far as I can see, or triactins at least, where you have one vertical ray pointing downwards, one pointing up, and then hooked over very sharply to project over the osculum. And then there's an extra ray pointing out and obliquely downwards. And these spicules also have this sort of sail-like structure of organic material around them as well. So that's a very different feature. It's unique to those spicules are unique to the oscular margin, and so you're starting to get a really complex and extremely interesting skeleton here. The transverse spicules between the fins along going up the body wall are relatively sparse. They're small, and it's difficult to see. I definitely agree with Vigby on that, but it does, I agree, also appear that they are transverse rays of spicules that were multi-rayed. So rather than being simple monaxons, there are some in this sort of area where you get a ray sticking up and then one going out sideways and crossing this gap between the fins. And there's actually really not much skeleton here. So that in itself suggests that the soft tissue of this sponge was actually fairly tough. It must have been able to support and maintain its own shape fairly well, because there's not much skeleton supporting it as far as we can see. What you have instead is this sort of the panels, the sheets between the fins, with very little holding it in place. And it would be difficult to understand that on the basis of modern sponges. This sort of gelatinous tissues you'd think would be too easy to collapse into the centre, especially if you're pumping water through it. So it looks like there is something else going on with the nature of these soft tissues that we don't see. In the same way as we get in ascosponges, these surfaced features that look like an epithelium, which sponges no longer have. Okay, now let's get to the symmetry, because another critical thing is that the description of having 8 fins is correct, making it octoradial. Almost. If you trace those fins down towards almost at the base, each pair of them fuse together into a single structure. So it's actually tetraradial, and then separates, each one separates into 2 to give you this sort of secondary octoradial symmetry around the body wall. And it is a real symmetry. It's constrained, it is consistent across every specimen that has been seen. This is a genuine octoradial stroke tetraradial symmetry, which of course sponges do not have today at all. I mean, octoradial symmetry is not a sponge feature. All the textbooks will tell you, and in fact university courses too, in biology and zoology, paleontology, that sponges are radially symmetric or asymmetric. They just don't have the genetic complexity, the control to mark out a true octoradial or tetraradial symmetric body plan. It's not in their nature. They also don't have any capability of secreting organic fins sticking out the side of the sponge. Yes, spicules today have a very thin outer organic layer in some cases, but we're talking microns. There's nothing that looks like a really thick structure that can then be embellished into really quite spectacular radiating sails down the side of it. There is kind of an exception in that the ascosponges again have these spicules with a thick organic outer layer, which was modified in some ways. There are some with little sort of ridges going up the side of it, but there's nothing like what we're seeing here. So we've got tetraradial symmetry, we've got spicular embellishment in a way we haven't seen before. We've got a body wall structure that seems to be much more self-supporting than you would expect from looking at modern sponges. In short, this thing doesn't fit anywhere in sponge evolution based on the modern groups. But there are a few echoes in other fossils. So tetravadial symmetry, for example, does actually turn up in quite a few early sponges, and in particular in a thing called Metaxispongia. This was described from the early Cambrian of China, one of the earliest assemblages of articulated sponges that we have, so just slightly older than Chengjiang. And these things contain 4 columns of diagonally oriented spicules running up the sides. And we see this in actually multiple specimens from different sites, multiple species, and you have between those you have panels of much more irregular hexaconid-based spicules, but they are clearly reticulosum-like sponges, thin-walled, conical shape, but they have this unique feature of 4 columns of spicules, which are in their own orientation. There are a variety of other species that we found in the Hertang biota from the same age, which have been illustrated again in a paper where I was looking specifically at this tetravadial symmetry. But we only got 1 or 2 specimens of each, and they were mostly slightly imperfect, so they've not been written up properly, they've not been formally described and named, but they are clearly there. And some of them are really quite interesting. There's one in particular which shows these columns of spicules are actually of asymmetric spicules. They are looking a lot more similar to things like those hooked ones on the top of the oscular margin, and also potentially these ones that cross the gaps between the rays in Takakkawia. So there's actually quite a lot of complexity in this group. There's a reasonable diversity of them in the earliest sponge faunas that we have, and then they basically disappear. Takakkawia is a leftover, and that is the youngest one of this group with this very obvious tetravadial symmetry that we see. There are traces of tetravadial symmetry later though, so the dictyosponges, for example, mainly from the Devonian of New York State, include quite a number with a very clear square form, or tetravadial form with sort of 4 projections or basically a square outline. There's a thing from the Burgess Shale, a reticulosum called Photoprisma, that has an 8-sided outline. And even if you go to Cyathophycus, one particular species of it we think was very close to the split between hexaconelids and demosponges, see the video on demosponges for that one, then that one, if you go to the earliest growth stage you can find, it has 8 spicules around the circumference. So there is actually quite a lot of evidence for tetravadial symmetry in a lot of these early sponges. But unfortunately, all these ones from the Hertang biota and Metaxispongia from the Huangbailing biota, they are all preserved just as the mineral components of the spicules. We don't see any trace of the organic embellishments that may or may not have been there. They're just not preserved. It's mainly pyritized, so we have pyrite replacement of the mineral phase, but not the organic part. Just the way it is. We're going to have to get some better ones. So, trying to summarize what Takakkawia actually looked like. We have a sponge, and there are these similarities to things like Metaxispongia in the overall structure, and it has spicules and so on, so there's little doubt that we should be calling it a sponge, I think. But we have a sponge that has utterly unique and divergent characters from everything else that we know about. These embellishments of the spicules produce the fins, the very, very, very clear tetravadial symmetry modified to octavadial, the unique spicules around the oscular margin. And it's also worth noting that those ridges on these little fins may be getting close to the spacing that is used as a natural diffraction grating to give you iridescence. So these things may have been utterly gorgeous to look at. You can imagine the sunlight scattering through the water and hitting these things and them basically turning all iridescent, a bit like the lines of cilia, or the comb rows on a ctenophore, for example. To pick an example completely at random, oh yes. So based on all those features, we cannot fit this into modern sponges. It can't fit anywhere near any of the living classes, and it can't really fit in the crown group. I mean, if that's a crown group sponge, then either large numbers of things have been completely lost in every other lineage independently, or it's somehow evolved tetraradial symmetry and all these other aspects as well. So I don't really buy that personally. It makes far more sense for this to be in the stem group, possibly a long way down the stem group of sponges. It's worth pointing out here that most people who work in this field or are publishing a little bit in this field would totally disagree with that, because the normal assumption is that sponges secreted spicules independently in every class. So to have a stem group sponge with spicules, well, that would have to be explained away as they evolved spicules independently as well, despite the fact that we have this sort of continuum with Metaxispongia, which looks very like reticulosans but with extra features, and yeah, it starts getting very contrived, and you have lots of things developing not just spicules, but similar architectures and arrangements of similar spicules in different groups independently. So we start to see difficulties with that. But anyway, moving on. If this is a stem group sponge, then you start noticing that things like tetraradial symmetry and octaradial symmetry are actually very widespread amongst other closely related phyla, ctenophores and cnidarians. It's complex. I mean, ctenophores are biradial technically, but they often approximate to octaradial symmetry. Cnidarians are generally tetraradial as adults. Yes, their larvae can be bilateral. It's exactly how the evolutionary relationships of these things work in terms of the symmetric homologies is difficult to work out, but tetraradial and octaradial symmetry are widespread in these other early branching phyla. So could we be seeing a relic here? Could this be something that is actually shared with the other phyla? Well, that would be interesting, wouldn't it? At this point I have to go off on a complete tangent and show you an early ctenophore fossil. This is a thing called Thoractina. It's from the Chengjiang biota, so very slightly younger than Metaxispongia and a bit older than the Takakkawia, but same sort of time interval. And Thoractina is part of a series of related fossils with different forms but similar structures that have been described from particularly the Chengjiang biota as sclerotenophores. They have comb rows. They have rows of sort of transverse bars which are supporting the cilia, which are the feeding apparatus of ctenophores today. But these are very different from modern ctenophores. And one of the features that they have is hints of a skeleton. They seem to have spicule-like structures in places which seem to support the body wall in different ways. And Thoractina is a really interesting thing, because in overall form it looks remarkably similar to Takakkawia. It has the same eightfold symmetry. It has these panels between them with the transverse bars. It has the little sort of flared structures around the apex. It has a little sort of the basal part sort of narrows, and that is very similar to the basal part of Takakkawia. And unfortunately we don't have the same level of detail of preservation, or at least it hasn't been published for Thoractina. So we can't compare whether we've got exactly the same sort of structure of the sort of the spicule arrangements that we see in Takakkawia, but everything that we can see matches up really surprisingly well. However, this and a later genus called Galeactina appear to have a statolith. At least this is a structure that it has been interpreted as. And statolith is a balance organ that is used by ctenophores that swim. Not all ctenophores swim around. There are benthic ones, and it's thought that the ancestral ones were benthic. But these ones have this structure at the tip, which would be the base in Takakkawia, because you have to turn it upside down, which looks like, or has been interpreted as, the statolith. Okay, there is, from everything that we can see here, remarkable similarity between these two fossils. And it's not quite watertight. We don't have all of the fine details we want to see. We don't see any of these little fins, potentially. Well, if we see fins, but we don't see that those fins are constructed in the same way as you get in Takakkawia. But I wouldn't bet against it, because we simply don't have the fine detail to show that that is what's going on. It's very likely, in my view, that that is exactly what's going on, because everything else about these is so similar that it's very hard to just assume that the one is a sponge, the other's a ctenophore, and they are nothing to do with each other. So if we take this a step further and allow the possibility, or at least investigate the possibility, that these could actually be, as they appear to be, closely related organisms, what does that mean? It means that the last common ancestor of sponges and ctenophores probably looked something like Takakkawia. Now, does that mean that they are sister groups? It does not, because what we're looking at is morphology. We're not looking at the pattern of evolutionary branching. So it's quite possible to have a last common ancestor of sponges and ctenophores, which also happens to be the last common ancestor of all other animals, closer to either the ctenophores or the sponges, but that's a higher node up the tree. It's also possible that that deep node between sponges and ctenophores could have looked rather like these things that are interpreted as early ctenophores stroke early sponges. However, that would depend on our phylogenetic interpretations of all of these fossils. And this first paper, presented by O et al., which described Thoractina and made this connection between various groups of Chengjiang things and coined the Ctenophore idea, also included a phylogeny. And in this phylogeny, the Ctenophores fall within the crown group of modern ctenophores. They are a branch within them that evolved probably relatively late within ctenophore evolution and diverged and then went extinct. And okay, if you believe that, it doesn't make any sense for the idea that there's any link between that and Takakkawia, because we would have to have the stem group of sponges evolving from within ctenophores, and we would have found, I would imagine, sponges falling within ctenophores in terms of their genetics, and we don't see anything like that. There's a lot of debate over whether sponges or ctenophores came first, as in was the first branch of animals, and it seems that they were very close together, but there's not a slightest hint that sponges fall inside ctenophores. So that would basically mean that this is a completely spurious comparison. They may look alike, but they are nothing like each other, really. Okay, however, let's have a look at this cladogram, because it does depend critically, well, all cladograms depend critically on the data that you put in. And one of the most critical pieces of data that you put into these analyses is the outgroup, because this is what you are using to polarize your sequence of character acquisition. So if your outgroup is one thing, then the most basal members of that group should converge with that outgroup. They should share quite a lot of features, or lack of features, with the outgroup. If your outgroup is something else entirely, then it can give you a different answer and swap around chunks of the tree to then have those closer to, or have members of the tree that are closer to the different outgroup being near the base, if you see what I mean. So the choice of outgroup makes a big difference to the arrangement of branches in the tree. And in this case, their outgroups are Eoandromeda, which is an interesting thing. It's a very curious structure with eightfold sort of spiral arms on a disc-like fossil, which is known from the Ediacaran, and has been suggested by some people to be ctenophore, but I don't think it's a very compelling argument. I don't think this is widely accepted as ctenophore, really, at all. And it's got this sort of 8 rays, but that's about all it's got going for it. The rest of it doesn't make sense, especially as they're sort of spiraling rays rather than just sort of having octaradial symmetry. So that's an odd one. It may not be a ctenophore, it may not even be an animal, we just don't know what that thing is. And then they have another outgroup which is labelled as hog. And I sort of scratched my head a bit and looked around the rest of the paper and couldn't find any reference to what the hog was. So eventually I found the supporting online information where all of the background data was, and it turns out that the hog is a hypothetical outgroup. So they have defined a set of features, a set of characters for a hypothetical outgroup which they think matches what this organism should have been like. And let me just read you what that listing was. In the Cladistic analysis, a diploblastic, soft-bodied animal with oral aboral axis, pelagic life habit, and uniformly ciliated body surface, hypothesized as the ctenophore ancestor, was used as a hypothetical outgroup. So they have chosen as an outgroup to define it. Not even used a real organism, it is something that they have imagined to be the outgroup, or to be a sort of a sister to this, to the ctenophores as a whole, having a uniformly ciliated body, so no comb rows, just cilia all over, pelagic, and with an oral aboral axis, but presumably presumably just radial symmetry. They haven't said whether it's tetraradial or anything like that, and that's what they've used as the outgroup. So it's no surprise that they get a pelagic, a soft-bodied thing with oral aboral axis, and otherwise being rather similar to their hypothetical outgroup, as the earliest branch on their tree of ctenophores, which then pushes the sclerotenophores further up the tree, because they are very, very different from their hypothetical outgroup. So in that case, you're actually, for whatever reasons that you have for choosing that as your outgroup, you have then dictated effectively what's going to happen in the rest of the tree. Okay, that was one cladistic analysis. There has been another one, so let's go back to that one. So the scenario of Thoractina and these other early sclerotenophores has been expanded actually by Zhao et al. In 2019 in an even bigger paper, which included a whole lot of other problematic early fossils, so things which are vaguely ctenophore-like or had never really been suggested to be related to ctenophores before, and has incorporated these into an even longer framework of early ctenophore ancestors. For those with a bingo card handy, it includes Deinonychus and Xiangwangia, so things which have long been problematic organisms within the Burgess shale-type faunas. Burgess and Chengjiang and others are turning up in this revision as part of an evolving sequence that fit in below the sclerotenophores. And they also have produced a major cladistic analysis, an extremely comprehensive one, they say, which seems to have been very well done, and you end up with a situation in which the sclerotenophores are not crown group ctenophores, they're the stem group. They're not as low down in the stem group as things like Deinonychus, but they are in the stem group of ctenophores, which makes it potentially more plausible to have a link with Takakkawia there. However, even that is a problem, since at the moment the genetics always seems to find ctenophores as the first animal phylum or sponges as the first animal phylum, and never sponges plus ctenophores coming off as a single branch together. And that would be required if these other organisms further down the ctenophore tree as they reconstruct it here are truly in that position and lower down the stem group. So it would mean that if these really are stem group ctenophores, then Takakkawia would also have to come off as a sister group to ctenophores. So sponges and ctenophores would have to form their own branch, which I don't have any philosophical problem with based on the fossil record. It's really a question of that's not what the genetics tends to find. And I must confess this is something I don't understand as to why that is the case, because it's always one or the other, pretty much always, and usually there's quite a small difference in age between which branch comes off first. And the difference between that arrangement and that arrangement seems to be quite subtle, and I'm surprised it doesn't turn up more often. But it doesn't. And so we have to sort of accept that based on the genetics, it seems unlikely that this precise arrangement is actually compatible with having the Thoractina and Takakkawia being closely related. Hope that makes sense. Because now I'm going to undermine that one as well. Because this tree, in particular, has at its base sponges as the first animal group. Which, okay, that is a point of view, but a lot of the modern work on genetics seems to be steadily converging, I think, towards actually the ctenophores being the first animal group and sponges shortly afterwards. And if you swap those around, then that might change things a little. So some of these early branches of ctenophores in this tree, like Xiangwangia, have previously been considered to be closer to the cnidarians. And they certainly do have some cnidarian-like characters, even though the authors of this paper have identified various features that they say are more diagnostic of ctenophores. But if you start having some of these organisms potentially smeared out on the tree towards cnidarians, and then you lose some of those ctenophore diagnostic features in that branch leading up to cnidarians and bilaterians, then theoretically not all of these need to be on the ctenophore branch. Now that is not the best explanation of the data that these analyses have found, but I'm not sure what would happen if you were to, for example, dictate that ctenophores had to be the first branch, and then include stuff like Takakkawia and Metaxispongia, code them up, and include that in the cladogram as well. I don't know what that would do. And that's the thing with these cladograms. They are always, no matter how thorough and how carefully done, they are always dependent on the data that goes into them. And you can tweak the data, you can add a few extra things, and suddenly you can induce large changes in the trees. So should we trust cladistic analyses implicitly? Not for giving us the answer. What we should trust them to do is to tell us the extrapolation of the data that we put into them. In terms of, if we have this set of characters and character combinations that we consider to be the important ones that we have included in the analysis, then that means that the most likely scenario for their evolution is this. It is definitely an if-then. It's not you do a cladogram and then you have the answer. It's very much a model, and the models are dependent on the things you put into them. So do I trust this one by Zhao et al. To be absolutely correct? No more than I trust any cladogram to give you definitely the correct answer. And when it doesn't include things like Takakkawia, and it does have a different topology of branches from what is now starting to become probably more just possibly slightly more consensus towards ctenophores first than sponges. I think it's still up in the air. But the thing is, we don't know at the moment. There are too many uncertainties around this, and one cladogram does not give us the answer. Nor does it doesn't, to be honest. But we work with what we've got, and these should always be seen as the outputs of models with whatever inputs went into those models. So, and it's a really interesting paper, and I do think they've got some very solid points about a lot of these features in these very early problematic groups potentially aligning with what we see in other early ctenophores. I just don't take the absolute answer of the phylogeny at face value, and neither should anyone else. Okay, so the moral is that I'm not sure where Takakkawia fits in animal evolution. I'm not even 100% sure that the similarity to Thoractina isn't a coincidence. I'm not 100% sure that it isn't actually on the ctenophore branch if it is a real similarity. So it's possible this is actually closer to ctenophores than sponges, but has all these sponge-like characters if they sort of blend into each other. But I don't know that for certain. All we can say is that it's an extremely interesting fossil that has often been overlooked in these discussions of early animal evolution. And if pushed, I would say that I think it's going to be very important for reconstructing the morphology of the last common ancestor of sponges with other animals, and therefore potentially important for reconstructing the last common ancestor of all animals. And one thing I'm absolutely adamant about is that this doesn't deserve to be ignored and omitted from these analyses, because it's an absolutely incredible critter. If this is a sponge, and with these other connections with Metaxispongia and so on, it's hard to argue that it's not at least very close to early sponges. Then it is so different from modern sponges that this thing has to be telling us about the ancestral conditions, the ancestral morphologies of the things that eventually became sponges. It's yet another line of evidence that sponges are secondarily simplified from a form which was much more like the polyps of cnidarians or the body form of early ctenophores, and it may be one of the closest fossils we have to one of these critical, really deep nodes in animal evolution. So there you go. This is why I've been steadily working up to Takakkawia, because it's an amazing thing. And not only does it help to sort of polarize evolution of the earliest animals, it also gives us sort of a bit of a baseline for the evolution of sponges beyond after this point, because so many of these features cannot be fitted into the crown group of sponges. I don't see any way that this can be a crown group sponge. So it's a really, really fascinating thing, and deserves to be a lot better known. And it's not the only weird one, and it's not the only weird one that might show some similarity to ctenophores. So at some point I'll have to tell you about Petaloption as well, but not today. That's quite enough headaches for now. Thank you all again. I will see you again soon, and please do keep writing those comments, and I will keep replying to them as much and as quickly as I can. Honest. Alright, bye bye.